Okay, today is our first chapter on the nerve cell and nerve impulses. Many of you may have not have had much discussion about this beginning material, so we're going to certainly lay it out as an introduction. So the nerve cell, the basic nerve cell is really called a neuron. You have billions of neurons in your brain, not just one neuron. So those neurons collectively work together to give you your experiences in life, to make you adapt to your surroundings. So at the basic level of studying the cell, I'll draw a little bit to help you out, but the images are there on the PowerPoint. I'm not going to go and test you on every single PowerPoint, but it's just a matter of you understanding the nature of how the nerve cell works. That's what we want. So for basic structure, the nerve cell actually looks like a tree. So that's a neuron. One. Again, you have billions of them. That little dot right there, nucleus. So in the nucleus is where you have your ribosomes, the DNA, the genetic material to make that neuron. So the nucleus becomes very important. Those extensions are called dendrites. So dendrites are the receiving portions of a cell. The portion that sends it out is called the axon. Axon sends, dendrites receive. And at the very end, there's looks like a little boot, a little foot. That's called the presynaptic terminal. So pre, that means there's something on the other side called post. This is pre. On the other side is post. What's in the middle of the pre and the post is something called a synapse. The synapse is like the gap, the space between neurons. Multiple neurons, but the space in between becomes important because that's where the chemicals flow through. Chemicals. So in this presynaptic terminal, oops, I erased the whole thing. There's something called vesicles. The vesicles contain the neurotransmitters that are released into the synapse that then goes to the postsynaptic neuron to talk to receptors. So that's a receptor. So when we look at these PowerPoint slides, you'll see those same images. I just wanted to visualize you being able to draw it yourself rather than just seeing an image. So beyond the neurons, you have glial cells. Glial cells are like supportive cells. You have 10 to 1 glial cells to neurons. So you have many more glial cells than you have neurons in your brain. So we speak of these neurons, brain, cerebellum, spinal cord. This is about 12 to 15 billion in the cortex, about 70 billion in the cerebellum, and about 1 billion in the spinal cord. Collectively, they work together. So we have, data from researchers in the past that have looked at the cellular structure. We talked about the nucleus and those very important elements inside the cell to make it work right, to have power to continue on. I think you remember from biology class, the mitochondria is for the powerhouse energy. You'll see some slides that has actually electromicrograph images of the cell cut, and you can see those parts of the nucleus, mitochondria, you get the ribosomes, and you got these very tedious, small structures that are key for that nerve cell to continue on to be what it's supposed to be. So when you look at the shape, it can be multiple shapes. I just drew one for you, but here's a neuron with its long axon, and these dendrites are coming off of it. Sustained neuron. So in your notes, you could have a motor neuron, a sensory neuron, and an interneuron. Here is discussing what a motor and the difference between a motor and a sensory neuron. Motor, certainly dealing with movement, sensory, dealing with smell, taste, touch, sensations. The shape's a little bit different. Motor neuron, here's a neuron, again, like a tree-shaped structure. The dendrites on the outside, nucleus in the middle, long axon, going to the what? Muscle, motor neuron. So around the axon, this purple stuff is called myelin. Myelin is the covering around the axon, like around a cord, some appliance that has wire in it, but around it is like insulation, or is a rubber covering. That covering is just like the myelin in the nervous system. And it doesn't cover it straight, but there's sometimes these little marks or little crimps, little spaces in between, and we'll talk about that a little later on. It's called the nodes of Ranvier. Sensory neuron, again, tree-like shaped structure. Here's the brain right here, or the central part of the sensory neuron, nucleus, soma also means body. You got the axon that sends the information. You got dendrites receiving over here. And so basically the skin is receiving information, processing, and going to the brain. So sensory neuron, motor neuron, interneuron just means a neuron that's pretty much in the spinal cord that can connect multiple neurons together. So from the head going to the spinal cord, and thus then get to your arm. So there's multiple interneurons that are located within your spinal cord. So we just discussed this again. Just know this for the exam. Dendrites, axon, soma. What we mean by the pre-synaptic terminal, we discussed that. Again, dendrites, read up what that means. Soma, what it means. Axons, what it means. And I'm going through this quickly because you also have the PowerPoints. So there's different shapes of neurons, just like there's different shapes of trees. So those neurons are differently shaped because of functions. So here's some diverse shapes. One was a bunch of dendrites, one long axon. This one has a sensory neuron, has multiple somas connected to these axons here. You got these that may be pretty much maybe associated with either the hippocampus or the cerebellum. They have long extensions. And then you have some that are very, very complex. They could be maybe the cortex. So again, the shapes, purple indicates where the soma is, but the branches show how very complex it can get in your brain. So beyond the neurons, I told you there's glial cells. So glial cells are the supportive cells. Complex names, but try to remember this. Astrocytes, microglia, oligodendrocytes, Schwann cells, those are the glial cells. So when we say astrocytes, they're like supportive type cells. They help to maybe make sure that a blood vessel gets to brain area. It makes sure it's supporting the way that the structure of the brain is forming so that the brain can get some blood. So it's like supportive type cells. Microglia, they help to absorb debris. If you had a stroke and your brain now needs to be cleaning those cells up, so microglia help to do that. So waste material, viruses, fungi. And again, we just talked about Schwann cells and oligodendrocytes. And you notice, these are the two cells that produce myelin. Oligodendrocytes produce the myelin in the central nervous system. Schwann cells produce the myelin in the peripheral nervous system. We'll go over those systems a little later on. Radioglia are neurons that help out during embryonic development. So again, these are all supportive type cells and not neurons themselves. And then, as we looked at the shapes of the neurons, we looked at the shapes of glial cells. So oligodendrocytes, again, they're pink right here just to show that they're showing the covering around the central nervous system. Schwann cell right here is purple, is showing it's the covering around the peripheral nervous system. Astrocytes, and see, you said like supportive type cells, it's actually holding up this blood vessel to get to a brain part. It's like a scaffold for a building. When the building is being built, on the outside is a bunch of metal and wood planks, so the person outside can help build that building. Well, the scaffold is not the building, it's just being supported to help to construct it. So the same thing right here. So there's also something called the blood-brain barrier. Just understand that there's like these tight junctions between in the vessels to ensure that there is no negative chemicals that are getting into the brain. So the barrier is protective. I mean, smelling, tasting, through your bloodstream, multiple things can get into your brain, and that's problematic because the brain cells, if they die out, you're in trouble. So the barrier is to ensure that it does not have harmful chemicals getting in. And when you look at the structure of it, here's an example of some things that can get through, like fat-soluble molecules, glucose. You need glucose, it's sugar. You need that for the brain to work. And then there's some tight wall junctions, real small, to make sure that it keeps out toxic things. You don't want formaldehyde and methanol and these negative chemicals getting into the brain because they can cause some significant damage. So just know a little about the blood-brain barrier and in the context of it protecting the brain. So we know we need glucose. Without glucose, your brain doesn't have the energy it needs. You've seen this later on when we talk about learning, but there's a disease called Korsakoff syndrome, and it results from a lack of something called thiamine getting into the brain. Thiamine is vitamin B1. So that deficiency happens because the person is no longer eating properly, because people who have Korsakoff syndrome are normally alcoholics. They're consuming alcohol and they're thinking they're getting their nutrition from the alcohol, but they're not. So they're lacking nutrition. Vitamin B1 is very important for getting into the brain, just like glucose is. Now, the next discussion on the neural impulse is how is it that communication occurs within the brain. So communication really is by electrochemical communication. So you have electricity and chemistry operating in your body. So when we say electrical, we're speaking about charged molecules. So electrical or electro chemical. Communication, that's how the brain is working. The electrical part deals with the way that charged molecules are operating in your brain. What are charged molecules? Ions, chemicals with electric charge, such as chlorine, magnesium, calcium, potassium, sodium, molecules with a charge. The chemical part relates to how when those ions are being charged, it helps to initiate the chemicals to be released at that place called that presynaptic terminal. It helps the chemicals to be released to make your brain work. So what are the chemicals? Basically your neurotransmitters. So neurotransmitters, dopamine, norepinephrine, epinephrine, serotonin, acetylcholine. Those are the neurotransmitters. Without them working, you have some body problems. So the issue is, how do we now deal with this issue of the nerve impulse? Well, just like a wire, I can't show you the wire, but if you have a wire, the energy flows down this wire as electricity. In your nervous system, it's not that simple. It actually jumps at those places where I told you there's spaces in the myelin. Those places and spaces are called nodes or rhombi. We'll see them again. Again, you have the PowerPoints. I'm just trying to lay it out for you to understand this. So the speed of the nerve impulse ranges from about 1 meter per second to 100 meters per second. I mean, that's fast, quick. So it's kind of hard to imagine what we mean by how quickly it can go from, let's say, the foot to the brain. So the properties of nerve impulse control are well adapted to the exact needs of how it needs to be transferred in the nervous system. I mean, your eye is different than your ear, different than your taste buds, different than your skin. So the nerve impulses are going to be different. If you, let's say, cut your finger, may not hurt till you look at it. If you stub your toe, the pain may not come until a few seconds later because it takes a while for it to get to the brain. So there's things that are different in terms of how it is being transferred because of the structure of the nerve. So to understand the first part of electricity, look at this. So when we said ions, this covering right here is the membrane or the circle. This right here is the membrane of the cell. If you remember from biology class, you have a lipid bilayer. We'll see a PowerPoint slide that shows that. I want you to conceptualize this about the nerve impulse. Until electricity flows down that axon to get to the end of it, to talk to the presynaptic terminal to submit the chemicals, you're not going to have any activity. So the ions and how they flow in and out of the cell become very critical. On that lipid bilayer, there are some places that chemicals can flow in and also flow out. So when there's nothing going on at rest, this membrane has a potential called resting membrane potential. If you actually took an electrode and stuck it in there, you put the electrode inside, the measure the inside of the cell is primarily negative because of chloride ions along the membrane. Cl is chloride, can't see that, Cl. And then a large anion. It's a large molecule with a large charge, that's what you're saying. So anions and chloride ions make a negative inside. At rest, negative 70 millivolts. On the outside is another ion called sodium. Sodium. Chloride, sodium. So sodium is on the outside. So if you've heard of the term action potential happens when the potassium ion, the sodium ions rush in, chloride ions come out, and so the membrane now is going to change. The membrane itself, if it's positive on one side, negative on another, that means it's polarized. Positive, negative, it's polarized. It's like North Pole, South Pole. Now, when there's a change in the membrane, it becomes depolarized. So, again, basics. You gotta have some ions that are flowing. That's electricity. You eat ions. Where do you get it from your diet? If you put sodium chloride together, what do you get? Right, salt. There's also something in here called potassium. Potassium, it's a K. K is the symbol for potassium. It's also small inside versus sodium larger outside, but there's always a balance between the way that charges, likes repel, opposites attract. So we're dealing with that in terms of the science of the cell. So potassium is also inside to help offset some of the chloride ions, but when this membrane changes, that number, negative 70 millivolts, goes into a positive direction. So that's just the drawing. Now let's try to visualize how it is on the slides. So again, negative 70 millivolts, resting membrane potential. That membrane, which says the lipid bilayer, there's your lipid bilayer. That green area right there is the location where the ions can go in and out. It's a protein molecule. It's the unit ion channel. It's a place where, again, the chemicals flow in and out of that layer. Here's one layer, another layer. You got a phospholipid head and a fatty acid chain. So the chain there is water repellent and the outside water attractant. So it's structured so it doesn't just fall apart. So that's one layer like a cake, but I'm telling you that's that whole circle I drew. That whole circle I drew is that membrane right there. Those ions flow through the green area. I told you that you can take an electrode, put it inside there, and record it, and see what the numbers are. And I also told you about that spaces between the myelin, where this nerve impulse is actually jumping. It's not just going down the axon like the wire in an appliance. It actually jumps. Space right there, that space right there, that's where it's jumping. So here again, showing you some ions and the forces that are allowing them to go opposing one another. When you look at these images here, it's showing you what's happening along the chain of events going down an axon. How one's closed right here, this was open, so it's allowing the sodium ions to come in. This was closed, so it goes down the chain of events like that. So I drew a circle, you saw it. I showed you that little layered structure with the lipid bilayer at the top and bottom with the little green structure inside. Well, here is now another example of, here's the membrane, here's those channels. So this is outside, that's inside. So this is outside the cell, inside the cell. So basically we're talking about inside is where you have some changes going on when that sodium flows in. Well, guess what? Sodium flows in and that potassium is going out, but it has to somehow go back to normal. How does it go back to normal? By a sodium potassium pump. For example, if you have some flooding in your basement and the water needs to get out of the basement, you might need a pump to pump the water out of the basement because you're going up to get it out. So the sodium potassium pump restores it back to balance, puts the sodium on the outside and potassium back on the inside. So terms. You can read this, but I need to show you the figure to kind of explain what this means so we can get through this quickly. So at rest, negative 70 millivolts, nothing happened. When that membrane changes, there's something called an action potential. So now the number, there is negative 70 right here, 0, and then the positive numbers. So it's going in a positive direction when it goes this way. So action potential, and then this is all in a time frame of 1 millisecond. Fast, you can't even think about how quick that is. 1 millisecond. So time by the electrical potential. So this one action potential is what's causing the nerve to be activated. It then goes back down to rest, but it overshoots before it actually comes back to rest. This overshoot area is called hyperpolarization. So again, right here, the memory is becoming depolarized. You have an action potential. It's now coming back to rest. It now goes below the rest of the memory potential. And during this phase, it's called hyperpolarization. There's two periods in here called the absolute and refractory periods, and they pertain to that the nerve impulse is not going to fire at that moment, anytime until it comes back to rest. So there's two periods in there called the absolute or relative refractory periods. Don't worry, I'm not going to ask you too much about that, but you need to understand conceptually what's happening with the ions as this is going on. So the same image here, you can put these on top of one another. While this is happening, this is going on. In red right here, sodium ions, that's how quickly they are going into the neuron when this is happening. Potassium is a little bit slower as it goes out. So that's why the height is lower, because it's not as many going out. And then it takes a longer duration for it to get back to what rest. So sodium and potassium function differently in that context, and in that understanding of this, think about anesthesia. You have general anesthetics, local anesthetics, and they may function to alter these particular ions. And that's why some knock you out, some of them are just local, because they're working on different ion systems. So again, when you look at this and try to interpret, try to understand that the ions are important as electricity. For electrical communication, it's the chemistry that we'll talk about in the next chapter, or next chapter on synapses, kind of explain what's happening with the way that this electricity is actually stimulating those chemicals. So When we spoke about the anesthetics, again, things like Novocaine, they may be local anesthetics that block sodium channels. So you go to the dentist, inject your mouth, don't feel that pain in the area because it's blocking the sodium channels, so you can't feel the pain, versus another medication that may knock you out in your sleep. So for nerve impulses to work, it's really an all or none principle. All or none states that the amplitude and velocity of an action potential are independent of the intensity of the stimuli. What do you mean by that? Intensity. If you ever flush the toilet, you can flush the toilet with a pinky, you can flush the toilet with a sledgehammer. It doesn't matter. A pinky, sledgehammer, that's totally different in terms of the intensities, but it's just a little click, a mechanism, you got action potential. So it doesn't have to be a gigantic, intense sledgehammer or a teeny prick from your pinky. If you look at the lever on the toilet, it's just a matter of it being released, and now you have an all or none principle, where you have it firing. And you can read up on that. The refractory periods, we discussed that briefly, but you can read what that means on the PowerPoint slide. And as we conclude this part, the sheath, or that covering around the axon, again, in the central nervous system is oligodendrocytes that produce it. In the peripheral nervous system is Schwann cells that produce it. So as the myelin is covering the axon, there's little spaces in between. The spaces are called nodes or ranvier. So the impulse actually jumps at those spaces. And the term for how it jumps is called saltatory conduction. So imagery, again, it's the axon. The purple stuff is the myelin around it. The spaces where the nodes are. And the term for how it jumps down the action, how it jumps down the axon, saltatory conduction. You can read that on your own. Any questions, you can certainly ask. In terms of diseases, people who have multiple sclerosis, deterioration of cells. For some reason, it's the deterioration of the myelin. So it's a fickle disease. People who may suffer from multiple sclerosis on some first line of symptoms could be eye problems. One of the largest myelinated nerves in your body is the optic nerve. So a person could have retrobulbar neuritis, optic neuritis. They have problems seeing focal vision because that axon that's heavily myelinated, for some reason, it's being deteriorated. The eyesight can sometimes come back without a problem, but then it's like physical problems that could happen because you have axons, you have dendrites, and you have this myelin all over the body. So if for some reason, as an autoimmune disease, it's attacking its own cells, destroying that myelin, making the person's body not work properly. So again, that sheath is very important as a covering to ensure the body works right. If you had a cord, an appliance, toaster, oven, your computer, and you cut away the insulation on the outside and expose the wire, it would probably shock you, or the appliance wouldn't work right. So that's the same thing with the body. So at the end of the slides, kind of tells you what I explained to you in the early part of the lecture. And I just didn't want to get too caught up and lost in the book, caught up and lost in the PowerPoints, but focus on the things that I emphasized, and you'll be okay to understand the nerve cell and nerve impulse. Next, we'll discuss the synapse.